So exercise 14, we are going to imitate an outbreak of a disease. So why would we do this? Basically, we're trying to mimic conditions for transmission of a pathogen between people in an actual population. Things to consider: there are numerous vectors or objects that can participate in the promotion of a disease in our society. Basically, it's not necessarily just person-to-person contact. Depending on which pathogen you're talking about, sometimes it's enough to touch a surface and have somebody else touch a surface to transfer it from one person to another. So, endemic, epidemic, and pandemic. Endemic means a disease is constantly present to a greater or lesser degree in people of a certain class or in people living in a particular location. Basically meaning that it is isolated, either by situation or location. Epidemic is an outbreak of a disease in a particular region. So it's not all over the place, but it is spread in the region that you see it in. Whereas a pandemic is geographically spread all over the globe. We're not talking about something being local. Something like cholera would maybe be in one specific place, or even malaria would be in one specific place because the conditions are right for it. But COVID-19, which we all know is a pandemic, would be an example of a pandemic. So what we did, we took coins that had been contaminated with yeast. They were given to one person and that person handled them. And then a separate coin that was the control that had nothing on it was given to another person. The person touched various objects in the lab, shook my hand. Then the things that the person each touched were swabbed with cotton swabs, basically, the Q-tip except sterile. These were tested for the presence of yeast. It's pretty simple to actually do. It's basically a catalase test in a test tube, since yeast can actually break down hydrogen peroxide. You put hydrogen peroxide into a test tube, you take the cotton swab that you've swabbed on the contaminated surface, and you look for bubbles. There are no bubbles. There's no contamination. If there is, then there is contamination. Once the swabs are collected, you pinpoint which coins are contaminated. I'm going to tell you ahead of time, I did odd numbers and even numbers for the coins, and the even numbers were the ones that were contaminated. What that meant was, whoever picked the even coin got the contamination on their hands. We identified who was handling them and who was responsible for the spread of the disease. We knew which one was the control because the control didn't show any type of bubbling, and we knew which one was the diseased coin because when we swabbed, we saw bubbles in the hydrogen peroxide. Looking at the results, student 1 handled the contaminated coin, student 2 handled the control or non-contaminated coin. The results for the contaminated coin are as follows. This was from the handshake. This was hand-to-hand contact. And you can see that there are bubbles coming up in that hydrogen peroxide. From there, we touched a chair. Now, admitted, there are some big bubbles like you saw in the first, but there are also quite a few little bubbles coming up from the sides as well. Even on the swab, you can see that there are bubbles. The next thing touched was a file box. You can see a lot of bubbles. Now, I'm not sure why. It seems like there are more bubbles in the third touch than there are in the second or the first, but it ended up being a lot. It almost looked like it was a carbonated beverage, honestly. And then the fourth thing touched was the tabletop. By the time that we got to the fourth touch, there are bubbles. Don't get me wrong, but there aren't as many bubbles here as there are in the other three. In handshake and touch, there are bigger bubbles than there are in the third and the fourth touch as well. Meaning that as multiple things were touched, I had less and less bacteria or pathogen on my hand. Since our reaction is getting smaller as I go, I'm releasing or depositing pathogen, leaving less on my hand to deposit in the next or following touch. This is our control results. This is no yeast on anything. And when we did the swabs, none of them had bubbles. None of them showed any presence of yeast because the person that had the control didn't have any yeast on their hands. Looking at the results, it demonstrates the difference between somebody with contamination spreading a pathogen and somebody with no contamination not spreading a pathogen. Literally looking at the cotton swab and looking for the presence of bubbles tells you that if there's contamination on something, it's easier to spread than if there isn't.